In this video, we'll be having a look at epithelia and looking at the coverings and linings associated with the epithelium. So one thing to consider is epithelium is the sort of that junction point between the outside world and underlying tissues, or it lines inside cavities and spaces. So it's that perfect spot for transportation or controlling what will enter into the deeper tissues. So, transportation across epithelia occurs by two routes. One is paracellular transport, and in this case, the substances leak between the cells. Now, of course, we have tight junctions, so that really limits what can come and go in most cases. So we have the paracellular route, so that means between the cells. And then we have the transcellular route, which means goes through the cells. And in this case, you have to cross the plasma membrane at one side. You have to work your way across the cytosol, diffuse across cytosol, and exit the cell, the plasma membrane, at the opposite side. So here's just sort of a graphical representation. Paracellular means that you're small enough you can actually work your way through there. Whereas transcellular, once again, you have to get through a cell membrane, work your way through across the cell to the other side, and come out the other side. Now, when we're looking at epithelia, we had looked at simple, but let's talk a bit about stratified. Now, stratified is the other big class of epithelium, and of course it means more than one layer of cells. And these are really good protective barriers. And they really are subjected typically to a high level of mechanical stress. Now, even under stratified epithelium, we can have different types. So one is keratinized stratified epithelium. And in this case, the apical cell layers are dead, it lacks nuclei, and it's full of this protein called keratin. And keratin is a very strong protein. So what happens is, as cells, basically, I'm just going to zoom in here. A great example of this is actually your skin. And what happens is the cells down here are the living ones. They divide, they push the older cells up, and as the older cells go up, they're going to transform. And by the time they get out here, you can see that they've kind of lost their nucleus, but they're full of, each cell's just packed full of this tough protein keratin. Right? So filled with keratin, that's a protein. It makes the tissue tough and resistant. And of course, this is what we see in the outer layers of our skin. Now, let's look at another type. Of course, you're probably starting to get the gist that if we're going to call something keratinized, stratified epithelium, there's probably ones that are not keratinized, or else we wouldn't have said keratinized. And you're right, is that we have a group called non-keratinized stratified squamous epithelium. In this case, the apical cells retain the nuclei. They're still alive, and they're in regions subjected to stress where most surfaces must remain moist. So that's an important point. Where do we find this? Well, look at some of the examples: the mouth, the throat, the esophagus, the anus, the vagina. So these are areas, yes, there is stress there. There is a lot of mechanical forces, but we don't want the same type of stratified squamous epithelium we have on our outer skin, which has all those dead, really tough armored cells, because of the nature of the location of the non-keratinized. We don't want it to be keratinized. But it's still, all these layers do give you a fair degree of protection. And you can see even out here in the outer layers, the presence of the nuclei. Now, we looked at stratified epithelium. Now let's look at some of the variants of it. We have stratified cuboidal, right? And we don't find this too often in humans. It's not one of the most common ones. It's two cell layers, and it lines things like ducts of sweat glands. And we can see it here is that you can see that it's two-layered, very typical cuboidal cell, so it is stratified. And of course, we also describe the shape of the cell as part of the name. Remember, we always start off with, is it simple or stratified? And the second part of the name will always sort of give us a description of the cell shape. And there is stratified columnar epithelium, also not that common. The apical layer is columnar, and the basilar cells tend to be cuboidal. And we can see that here. These cells tend to be more kind of cuboidal in shape, whereas the apical ones are a bit more columnar in shape. And once again, this is associated with things like salivary glands. Now, there's another group called transitional epithelium. Now, this one, once again, is not a fairly unusual one, and only we find this in the urinary system. So it lines part of the inside of the kidneys, the ureter, the urinary bladder, urethra. And here the cuboidal basilar cell layers, and then we have a dome-shaped apical cell layers in the relaxed tissue. But the tissue, like think here in the bladder, right? Your bladder, when it fills, will expand out, and the tissue gets stretched as it does. So not surprisingly, that this tissue that looks like this, in a relaxed state, when you stretch it out, you'll start to see that the surface cells get kind of pulled and flattened out. And that's why it's called transitional, because the tissue changes looks, whether the bladder is empty and the tissue is not being stretched out, or whether the tissue is stretched out. And here's just a great summary. And as I said, you know, the book has great summaries. And remember, get in there, read the book, have a look at the content, and really use all your resources to try and get a good sense as to what is happening. So we're going to end the video here, and the next one we're going to come back and talk about glandular epithelium.